the Yukagir woolly mammoth. In 2002, along a remote bend of the Matsunuoka River in northeastern Siberia, hunters searching for ivory noticed a shape emerging from the thawing permafrost. It was not stone. It was not fossilized bone. It was a curved tusk attached to something far more unsettling. Beneath layers of frozen sediment lay the head of a woolly mammoth, its skin still intact, its features still defined. The Ice Age had not reduced it to fragments. It had preserved it. The specimen, later known as the Yukagir mammoth, was dated to roughly 20,000 to 25,000 years before present. What made the discovery extraordinary was not the age, it was the condition. The head retained patches of coarse reddish-brown hair several inches long. The leathery skin had not mineralized into stone. Muscle structure beneath the surface could still be identified. Even the contours of the trunk base and cheek were visible. This was not the usual image of prehistory reconstructed from scattered bones. It was an animal halted mid-existence. Under normal circumstances, a body the size of a mammoth retains internal heat long after death. That residual warmth allows bacteria to multiply, soft tissue to liquefy, and scavengers to dismantle what remains. Complete preservation of skin and hair in a creature weighing several tons requires something different. It requires rapid and sustained freezing before decomposition can progress. Permafrost provides that environment, but only if burial and temperature conditions align precisely. The Yukagir mammoth suggests that alignment occurred with remarkable efficiency. Examination of the surrounding sediment showed frozen silts and fine-grained deposits consistent with Ice Age steppe tundra. Pollen and plant fragments embedded nearby pointed to a cold, grassy ecosystem. Some mammoth specimens discovered elsewhere have contained undigested plant matter in their stomachs, including grasses and flowering plants from temperate seasons. While not every carcass preserves internal contents, the broader pattern raises a recurring scientific question. How quickly did these animals transition from living organisms to frozen archives? The Yukagir head required careful extraction to prevent damage once exposed to warmer air. As permafrost thaws, tissue that has remained stable for millennia can degrade within hours. Specialists stabilized the specimen and transported it under controlled cold conditions for further analysis. Studies focused on morphology, hair structure, and the potential for recovering genetic material from preserved tissue. Even fragmented DNA from such remains can illuminate evolutionary relationships between mammoths and modern elephants. Yet the physical presence of the specimen communicates something beyond laboratory value. Standing before a preserved mammoth head is not like observing a fossil cast. The texture of the hide, the direction of the hair, the curvature of the tusk create an immediate biological reality. This animal once moved across a windswept plain. It breathed, fed, migrated. Then, at some point, it entered a chain of events that ended with its body locked into ground that would not thaw for tens of thousands of years. Climate records from ice cores show that during the last glacial period, temperatures in parts of the northern hemisphere fluctuated significantly. Some shifts occurred over centuries. Others appear in the geological record as more abrupt transitions. Whether the Yukagir mammoth died during a gradual cooling phase or in the midst of a sudden environmental change remains debated. What is not debated is the evidence of rapid freezing relative to decay. The permafrost did not simply bury bones. It captured tissue before time could erase it. If a multi-ton animal could be preserved with skin, hair, and structural details still recognizable after 20 millennia, then the cold that sealed it in place was not ordinary. It was decisive, and the deeper the Arctic thaws today, the more such decisions from the past are beginning to surface. The baby mammoth Lyuba. In May 2007, along the Urabe River on the Yamal Peninsula in northwest Siberia, a reindeer herder noticed a small, dark form exposed by melting riverbank ice. At first glance, it resembled the body of a recently deceased animal. It was less than 4 feet long, its limbs tucked close to its body. The skin was intact. The trunk was still formed. This was not an adult mammoth reduced to fragments by time. It was a baby, later named Lyuba, preserved with a level of completeness rarely seen in the fossil record. Radiocarbon dating placed Lyuba at approximately 42,000 years old, yet the body showed no signs of the extensive decay expected over even a fraction of that time. The hide remained elastic, The ears, though slightly shrunken from dehydration, retained their structure. Even the tiny tail was present. CT scans later revealed that much of the internal anatomy remained in place. The skeleton had not scattered, the organs had not vanished. The body cavity was filled not with emptiness, but with sediment and preserved tissue. What distinguished Lyuba scientifically was not only external preservation, but internal detail. Imaging studies showed well-defined muscle layers and intact joints. The lungs and portions of the digestive tract were identifiable. Analysis of stomach contents indicated she had ingested her mother's milk shortly before death. In the intestinal tract, researchers found traces of plant material and microorganisms typical of steppe environments. This meant that the freeze occurred quickly enough to halt digestion mid-process. Under typical post-mortem conditions, bacterial activity begins within minutes. Autolysis, the self-digestion of cells, follows as enzymes break down tissue from the inside. In a body as small as Lyuba's, complete soft tissue decomposition would normally proceed rapidly unless temperatures dropped below freezing very soon after death. The evidence suggests that she likely died in a muddy riverbank environment, possibly suffocated in sediment, and was then rapidly entombed in cold conditions that stabilized the body before decay advanced. Further examination revealed another detail. The body cavity contained fine silt, consistent with floodplain deposits. This indicates that burial was not a slow accumulation of snow over decades, but a relatively swift covering by wet sediment, followed by freezing. The combination of rapid burial and sustained sub-zero temperatures created a natural cryogenic chamber. Once sealed in permafrost, microbial activity would have been effectively suspended. The microbial findings were as significant as the anatomical ones. Samples taken from Lyuba's digestive system provided insight into the gut flora of Ice Age megafauna. These microorganisms, preserved in frozen stasis, offered a biological snapshot of a vanished ecosystem. Unlike bones, which tell of structure, preserved tissue and microbes reveal function. They reveal metabolism, diet, and interaction with environment. Climate data from Greenland and Siberian ice cores showed that the Late Pleistocene was not a static deep freeze. It was punctuated by abrupt climatic oscillations. Some transitions between colder and warmer phases appear in the geological record over relatively short time scales. Whether Lyuba's death coincided with one of these shifts cannot be stated with certainty. What can be stated is that her preservation required a drop to stable freezing conditions before internal decomposition could fully unfold. Standing before the preserved body of a juvenile mammoth alters perception of deep time. The proportions are familiar. The curvature of the trunk resembles that of a modern elephant calf. The eyelashes are still visible. The illusion is temporary but powerful. It suggests that tens of thousands of years can be bridged not by imagination but by tissue. Lyuba does not confirm a single catastrophic theory. She does not demand an extraordinary explanation, but she reinforces a pattern established by other Arctic finds. Large mammals, and now even their young, were at times sealed into the ground quickly enough to preserve skin, organs, and microbial life. If a calf that had just fed could be buried and frozen before digestion finished, then the environmental transition from life to ice was not prolonged. It was immediate, and in the frozen ground of the Arctic, that immediacy is written in flesh rather than stone. The Yukagir steppe bison? In 2011, in the same vast Siberian region that had yielded mammoth remains, a new shape emerged from the permafrost near the Yana River basin. Unlike scattered bones typically recovered from Ice Age sites, this discovery had weight, form, and mass. It was the hindquarters of a steppe bison, dated to over 30,000 years before present. The muscle fibers were still visible. The hide still clung to the flesh. When cut, the tissue did not crumble into dust. It separated along anatomical lines. The steppe bison, an ancestor of modern American bison, once roamed the mammoth steppe in enormous herds. Adults could weigh over 2,000 pounds, built for endurance in open grasslands that stretched across Eurasia and into North America. In life, they endured extreme winters. In death, however, preservation of soft tissue on such a large mammal is statistically rare. Yet this specimen retained not only skin and hair, but defined muscle structure beneath the surface. Laboratory analysis confirmed that the collagen matrix within the muscle tissue had not fully degraded. While cellular membranes do not survive intact for tens of thousands of years, the broader architecture of the tissue remained identifiable. That level of preservation requires sustained freezing at temperatures low enough to suppress bacterial proliferation. Once temperatures stabilize below 32 degrees Fahrenheit and remain there, enzymatic breakdown slows dramatically. But for a body this size, internal cooling must occur rapidly to prevent early decomposition. The sediment encasing the bison consisted of fine-grained silts mixed with ice lenses, characteristic of permafrost floodplain deposits. Such environments can trap animals that fall through unstable ground or become mired in mud. If burial occurs quickly, oxygen access is limited. Without oxygen, many decomposing bacteria struggle to function. Combine that with an immediate drop below freezing, and decay can be halted before it gains momentum. What makes the steppe bison compelling is not simply that it was preserved. It is that the pattern repeats. Mammoths, rhinoceroses, horses, and now bison all display variations of the same outcome: large herbivores frozen with substantial soft tissue intact. In Alaska, similar discoveries have been made within deposits known as muck layers, where bones and carcass fragments are found intermixed with plant material. In some cases, muscle tissue recovered from frozen bison has even been described as structurally recognizable enough to study fiber orientation and composition. This repetition leads to a restrained but persistent question within paleobiology. Were these animals dying gradually across centuries of slow climatic deterioration? Or were some caught in episodes of sudden environmental stress? Ice core records from the Late Pleistocene indicate that temperature shifts in the northern hemisphere could occur over relatively compressed time scales in geological terms. These shifts do not necessarily imply instantaneous global freezing, but they do demonstrate that climate systems are capable of abrupt transitions. The Yukagir steppe bison does not prove a planetary catastrophe. It does, however, reinforce the physical reality that freezing conditions were established quickly enough in certain locations to preserve heavy muscle mass before it decomposed. A 2,000-pound body does not freeze uniformly from the outside inward without time playing a role. The fact that identifiable tissue remains suggests that burial and cold acted in coordination. When displayed in controlled environments, the preserved hide and muscle do not resemble a fossil cast. They resemble something that should not have survived the passage of tens of millennia. The surface texture is biological, not mineral. The weight is organic, not lithified. Each new specimen strengthens the same observation. The Arctic is not merely a graveyard of bones. It is a vault of bodies, some of them halted in a condition far closer to life than the word prehistoric implies. If repeated discoveries across Siberia and Alaska show large mammals preserved with skin and muscle intact, then the key variable is not age. It is the speed at which cold overtook them. And that speed, recorded in tissue rather than sediment alone, is what continues to draw scientific scrutiny toward the frozen ground of the north. The cave lion cubs of Yakutia. In 2015, deep within the permafrost of Yakutia in northeastern Siberia, miners searching for mammoth ivory uncovered something far smaller and far more delicate than tusk or bone. Inside a frozen riverbank, encased in compacted ice and sediment, lay the bodies of two cave lion cubs. They were not skeletons. They were complete. Their paws were intact. Their whiskers were visible. Their fur still covered their skin in fine detail. For tens of thousands of years, they had not seen light. Radiocarbon analysis later dated the cubs to approximately 12,000 to 40,000 years before present, placing them within the Late Pleistocene, near the end of the last ice age. Cave lions were apex predators, larger than modern African lions, adapted to the cold grasslands of Eurasia. Adults are known primarily from bones recovered in caves and open steppe deposits. But these cubs were different. They were juveniles, estimated to be only a few weeks old at the time of death. The preservation was extraordinary. The fur retained its pale tawny coloration. The paw pads were defined. The facial structure, including eyelids and nose, remained clearly formed. Unlike mummified remains that dry out over time, these bodies were held in a frozen state that prevented the collapse of soft tissue. CT scans revealed that internal organs had not completely decomposed before freezing stabilized the structure. Even the stomach contents and lung cavities could be examined. For a predator of this size, Early death likely occurred in or near a den environment. One hypothesis suggests that a sudden collapse of the den roof, perhaps triggered by soil instability or thawing and refreezing cycles, buried the cubs rapidly. Fine sediment surrounding the bodies indicates swift coverage rather than gradual accumulation. Once encased, the drop to stable sub-freezing temperatures would have halted bacterial decay before it progressed extensively. Under typical conditions, small mammals decompose quickly. Body heat dissipates rapidly, but so does structural integrity. Without freezing, tissues break down, fur detaches, and the body collapses inward. The cub's preservation indicates that decomposition had barely begun before environmental conditions intervened. Their posture suggests they were not scavenged. They were not exposed to prolonged surface decay. They were sealed. What distinguishes the cave lion cubs from large herbivore finds is vulnerability. These were not massive animals with thick hides and insulating fat layers. They were young carnivores with thin skin and delicate bone structure. If freezing had been slow or inconsistent, tissue would have deteriorated. Instead, their features were preserved with a clarity that allows scientists to study musculature, fur structure, and even aspects of developmental anatomy rarely accessible in extinct predators. Their discovery reinforces a broader pattern observed across Arctic permafrost sites. Mammoths, bison, horses, rhinoceroses, and now apex predators appear in similar frozen states. Each case involves rapid burial and sustained cold. The precise environmental triggers may vary, but the biological outcome is consistent: preservation before decay. Ice core records indicate that the terminal phase of the Pleistocene was marked by climatic volatility. Rapid cooling events and equally rapid warming periods are recorded in isotopic signatures extracted from Greenland cores. While these transitions unfolded over years to decades rather than hours, they demonstrate that Arctic systems can shift more abruptly than once assumed. In localized settings, such shifts combined with geological instability could create conditions favorable for sudden burial and freezing. The cave lion cubs are silent, but their condition speaks. They represent a moment in time when life ceased and the environment intervened almost immediately. Their fur did not rot away. Their skeletons did not scatter. Their bodies became biological records sealed beneath frozen earth. When examined under controlled laboratory lighting, the cubs do not resemble artifacts. They resemble sleepers. The illusion lasts only a moment before the scientific reality reasserts itself. They are extinct, separated from the present by tens of thousands of years. Yet the ice preserved them with such precision that the gap feels compressed. If predators as fragile as newborn cave lions could be buried and frozen before nature dismantled them, then the environmental conditions responsible were efficient and decisive. And in the Arctic, that efficiency continues to surface, one body at a time: the Tumat Puppy. In 2011, near the village of Tumat in northeastern Siberia, ivory hunters working along a thawing riverbank uncovered a small, fur-covered body emerging from the permafrost. At first glance, it appeared to be a recently deceased animal, perhaps a modern dog lost in the tundra, but the surrounding sediment told a different story. The layer belonged to the Late Pleistocene. The body was not recent. It had been sealed in frozen ground for more than 12,000 years. The specimen, later referred to as the Tumat Puppy, measured just over 3 feet in length. Its fur was dark and dense. The limbs were intact. The paws still bore claws. Even the whiskers were visible along the muzzle. Unlike skeletal remains typically found in archaeological sites, this animal retained soft tissue throughout much of the body. The hide had not mineralized. The musculature beneath the surface was still identifiable. Radiocarbon dating confirmed its antiquity. What made the discovery particularly significant was not only preservation, but context. The puppy was found near the remains of a mammoth bone structure, interpreted by some archaeologists as a possible human dwelling or activity site. Burn marks and cut bones had been identified in nearby sediments. This placed the animal in proximity to early human populations at the end of the Ice Age. The central question quickly became taxonomic. Was the Tumat specimen a wolf cub, a primitive dog, or something transitional between the two? Genetic analysis was undertaken to compare mitochondrial DNA with that of modern wolves and domestic dogs. Results indicated that it was closely related to ancient wolf lineages, but the broader debate over early dog domestication remains unresolved. The specimen sits within a period when wolves and early humans were already interacting across Eurasia. From a preservation standpoint, the Tumat puppy follows the same pattern seen in other Arctic finds. The body shows minimal signs of scavenging. The posture suggests it was not exposed for long before burial. Fine sediment and ice lenses surrounded the carcass, consistent with rapid coverage followed by sustained freezing. In a small mammal, internal heat dissipates quickly, but decomposition can proceed just as fast if temperatures remain above freezing. The state of the tissue implies that freezing stabilized the body before decay progressed significantly. CT imaging revealed internal structures that had not fully collapsed. Portions of muscle and connective tissue remain distinguishable. Unlike mummified desert remains, which dehydrate gradually, this preservation reflects cryogenic suspension. Once sealed in permafrost, microbial activity would have been dramatically reduced. The result is a body that bridges the gap between fossil and recent death. The environmental backdrop to the Tumat find coincides with a period of climatic fluctuation known as the Younger Dryas, an interval characterized by abrupt cooling in parts of the northern hemisphere. Ice core records suggest that temperature shifts during this time could occur over relatively short geological intervals. While the precise cause of the Younger Dryas remains debated, its existence confirms that rapid climate transitions were possible in the Late Pleistocene. The Tumat puppy does not prove domestication. It does not confirm a catastrophic freezing event. What it does provide is a biological snapshot from a transitional era when humans, wolves, and megafauna shared overlapping territories. The preservation of fur, tissue, and genetic material allows researchers to examine that relationship in unprecedented detail. When viewed under laboratory conditions, the animal's proportions are unmistakably canine. The curvature of the snout, the shape of the paws, the texture of the coat evoke familiarity. Yet, this familiarity is separated from the present by more than 10 millennia of frozen ground. If a small carnivore living alongside early humans could be buried and stabilized in ice before decomposition erased its features, then the conditions that sealed it were not prolonged and gradual. They were swift enough to intervene between life and decay. And in the Arctic today, as permafrost continues to thaw, similar interventions from the past are beginning to reappear. The baby woolly rhino Sasha. In 2014, along the banks of the Similyak River in northeastern Siberia, a hunter searching for mammoth tusks noticed a dark mass protruding from thawing permafrost. Unlike the massive silhouettes of mammoth remains occasionally found in the region, this shape was smaller, compact, and unexpectedly intact. When the surrounding ice was cleared away, the body of a juvenile woolly rhinoceros emerged, later nicknamed Sasha. It was estimated to be only a few weeks old at the time of death. Yet its skin, fur, and much of its soft tissue had survived for more than 30,000 years. Radiocarbon dating placed Sasha firmly within the Late Pleistocene. Woolly rhinoceroses were large, thick-skinned herbivores adapted to cold steppe environments, often weighing over 4,000 pounds as adults. But this specimen was a calf. Its body measured roughly 4 feet in length. The hide, though dehydrated in places, remained attached. Patches of dense underfur and guard hairs were still visible along the back and flanks. The ears, small and rounded, retained their structure. Even subtle details of the skin folds were preserved. Soft tissue preservation in large mammals is rare. In juveniles, it is rarer still. Young animals have thinner skin and less insulating fat. They are more vulnerable to environmental stress and microbial breakdown after death. Under ordinary conditions, autolysis begins within minutes, followed by bacterial proliferation that rapidly dismantles internal organs and connective tissue. For Sasha's body to retain recognizable musculature and dermal layers, freezing must have occurred before decomposition advanced significantly. Examination of the surrounding sediment revealed fine silts and frozen soil, consistent with rapid burial along a riverbank or floodplain. Such areas are prone to sudden collapse, particularly during seasonal transitions, when thawing and refreezing cycles destabilize the ground. A young rhinoceros could have become trapped in mud or shallow water, and then quickly covered by sediment. Once sealed beneath wet earth, a drop in temperature below freezing would have stabilized the carcass. CT imaging and external inspection indicated that the internal cavity had not completely decomposed before freezing. While some organs had degraded, structural integrity remained sufficient to identify anatomical features. Researchers examined hair samples to understand insulation properties and coloration. The hide thickness and collagen structure offered insights into how these animals adapted to glacial climates. Even partial preservation of such tissues provides more biological information than skeletal remains alone. Sasha's discovery reinforced a growing pattern across Siberia. Mammoths, bison, horses, cave lions, and now woolly rhinoceroses had all been found with varying degrees of soft tissue preservation. Each case pointed to a similar environmental sequence: rapid burial, followed by sustained freezing. The differences lay in species and age, not in the mechanism of preservation. The late Pleistocene climate was not static. Ice core data from Greenland and sediment records from Eurasia show repeated fluctuations in temperature and precipitation. While many changes unfolded over decades or centuries, the geological record confirms that shifts could occur more abruptly than once assumed. In regions with unstable permafrost and active river systems, even localized events could create conditions for swift entombment. Standing before the preserved body of a juvenile woolly rhinoceros compresses the perception of time. The proportions are familiar. The texture of the hide resembles that of modern rhinoceroses. Yet this animal lived in a world dominated by mammoths and steppe grasslands, long before recorded human history. Sasha does not require an extraordinary hypothesis to explain its existence. Rapid burial and freezing are sufficient. But the repeated emergence of such well-preserved juveniles strengthens the same observation. The boundary between life and permanent ice was crossed quickly. If even the most vulnerable members of Ice Age megafauna could be sealed in permafrost before decay erased their form, then the environmental forces at work were not slow and forgiving. They were immediate. And as Arctic ground continues to thaw, more of those immediate moments remain locked just beneath the surface. The Batagaika Foal. In 2018, within a vast thermokarst depression in northeastern Siberia known as the Batagaika crater, researchers encountered a form emerging from melting permafrost that did not resemble bone or driftwood. The Batagaika crater, often described as one of the largest permafrost thaw slumps in the world, stretches nearly half a mile in length and continues to expand as ice-rich ground collapses inward. From its exposed wall of frozen sediment, the body of a prehistoric foal appeared, suspended in earth that had not thawed for tens of thousands of years. The animal, later identified as a late Pleistocene horse, was estimated to be approximately 2 months old at the time of death. Radiocarbon dating placed its age at over 40,000 years. Unlike skeletal remains typically associated with extinct horses, this specimen retained skin, mane, tail hair, and significant portions of internal tissue. The hide was dark brown and unbroken. The hooves were intact. Even fine hair around the muzzle and legs remained visible. What distinguished the Batagaika foal from previous discoveries was the condition of its internal organs. CT imaging revealed preserved lung structure and remnants of the digestive tract. Muscle tissue had not fully disintegrated. In some areas, cellular architecture could still be discerned at a structural level. For an animal of this size, preservation of internal anatomy requires rapid cooling following death. Without freezing, enzymatic processes and microbial activity would have dismantled organ tissue within days. Sediment surrounding the body consisted of fine silts, typical of riverine or floodplain environments. One prevailing hypothesis suggests the foal may have become trapped in mud near a water source. Once immobilized, it could have been quickly covered by sediment during a flood event. If temperatures dropped shortly afterward, freezing would have stabilized the body before decomposition advanced. The combination of burial and cold created conditions similar to natural cryopreservation. Researchers collected tissue samples to assess DNA integrity. Although genetic material degrades over time due to chemical breakdown and radiation exposure, the extreme and stable cold of permafrost slows that process significantly. Preliminary analysis indicated that the DNA fragments recovered were sufficiently preserved to contribute to ongoing studies of extinct horse lineages and their relationship to modern breeds. Such material allows reconstruction of evolutionary pathways that skeletal morphology alone cannot fully reveal. The Batagaika crater itself adds another dimension to the discovery. The depression has grown rapidly in recent decades due to rising regional temperatures and ground instability. As permafrost thaws and collapses, layers of ancient sediment are exposed in vertical cross-section, effectively revealing a timeline of environmental history. The foal emerged not because it was newly preserved, but because the ground holding it had begun to fail. Late Pleistocene climate records show repeated oscillations between colder and milder intervals. While many of these transitions occurred over years or decades, the record confirms that environmental systems were dynamic rather than static. In such settings, localized conditions could shift quickly enough to trap and freeze animals inhabiting vulnerable terrain. The preserved foal presents a striking image. Its proportions resemble those of a modern horse. The curve of the neck, the alignment of the legs, and the texture of the mane appear familiar. Yet it belonged to an ecosystem populated by mammoths, woolly rhinoceroses, and cave lions. Its world vanished, but its body remained sealed beneath frozen ground for over 40 millennia. The Batagaika Foal reinforces a pattern now repeated across Siberia. Large and small mammals, juveniles and adults alike, were in some cases buried and frozen before natural decay erased their biological detail. The ice did not fossilize them, it suspended them. As permafrost continues to thaw and ancient layers collapse into view, discoveries like this are no longer isolated anomalies. They are becoming part of a growing record. If an animal that died over 40,000 years ago can emerge with hide, hair, and internal structures still recognizable, then the freezing that preserved it acted decisively. And with each collapse of Arctic ground, the question expands beyond what is found, toward what still remains sealed within the ice. The Yakutian cave bear. In 2020, reindeer herders traversing the Bolshoi Lyakhovsky Island in the Russian Arctic encountered a shape emerging from melting permafrost that appeared almost contemporary. The body lay partially exposed along a thawing coastal slope, its head intact, teeth visible through parted jaws. Unlike skeletal remains commonly found in caves across Eurasia, this specimen retained skin, fur, and soft tissue. It belonged to an extinct cave bear, a species that vanished approximately 15,000 years ago. Cave bears were massive omnivores of the Late Pleistocene, often exceeding 1,000 pounds in weight. Most knowledge of the species comes from bone accumulations discovered in European caves, where skeletal remains were preserved in dry conditions. Soft tissue preservation, however, had never been documented at this scale. The Arctic find altered that record. The hide remained attached across much of the body. The nose structure was intact. Even the claws were preserved at the ends of the paws. Radiocarbon analysis was initiated to determine the specimen's age, while external examination confirmed that decomposition had been halted at an early stage. Fur still covered large sections of the torso. The ears retained form. The snout and lips, though slightly desiccated, were recognizable. In temperate climates, a carcass of this size would decompose rapidly due to microbial activity and scavenging. In this case, freezing must have intervened before those processes advanced. Sediment surrounding the body consisted of ice-rich silts, typical of Arctic permafrost deposits. One plausible scenario involves the bear entering a den, or depression that later collapsed or flooded. Swift burial in wet sediment, followed by sustained sub-zero temperatures, would have stabilized the body. Once sealed in frozen ground, oxygen access would have been limited, slowing bacterial growth dramatically. The presence of preserved soft tissue in a large omnivore broadens the ecological pattern observed across Arctic discoveries. Herbivores, such as mammoths and bison, predators like wolves and cave lions, and now a dominant omnivore appear within the same frozen archive. Preservation is not limited to a single species or dietary niche. Ice core records confirm that, during the Late Pleistocene, Arctic regions experienced repeated cold phases interspersed with warming intervals. Permafrost expansion during colder phases could entomb organisms rapidly, particularly in unstable terrains near rivers, coastlines, or collapsing slopes. Once frozen, the biological clock slowed almost to stillness. The cave bear's preserved muzzle, with teeth still set in place, compresses the distance between prehistory and the present. The curvature of the claws resembles that of modern brown bears. The fur texture appears familiar. Yet the species itself disappeared at the close of the Ice Age, likely due to climatic stress and competition with humans. The discovery does not demand an extraordinary explanation beyond rapid burial and persistent cold, but it reinforces the same conclusion drawn from previous finds. Across multiple trophic levels and body sizes, late Pleistocene animals were in certain cases sealed into permafrost before decay erased their anatomy. If even a large omnivore such as a cave bear could be preserved with hide and claws intact for tens of thousands of years, then the Arctic permafrost functioned not merely as frozen soil, but as a stabilizing vault. And as warming temperatures continue to destabilize that vault, organisms once confined to geological time are re-emerging into a climate very different from the one that froze them. The Duvanny Yar Wolf. In 2021, along the banks of the Kolyma River in northeastern Siberia, erosion along a permafrost cliff exposed the body of a large canid embedded within frozen sediment. The site, known as Duvanny Yar, is a towering permafrost outcrop that reveals layers of Ice Age deposits stacked like pages of geological history. As seasonal thaw destabilized the frozen wall, a dark shape emerged from the ice. It was not bone, it was fur. The animal was identified as a late Pleistocene wolf, dated to more than 32,000 years before present. Unlike skeletal remains commonly recovered from steppe deposits, this specimen retained skin, hair, and significant soft tissue. The head was intact, the ears were still formed. The jaw, lined with sharp carnivorous teeth, had not disarticulated. Even the nose and whisker pads remained visible, compressed by time, but not erased. Wolves are highly adaptable predators. Modern populations survive in Arctic climates, yet the preservation of this individual suggests more than cold tolerance. For muscle, hide, and connective tissue to persist over tens of millennia, freezing must have stabilized the body before autolysis, and scavenging could dismantle it. A predator carcass left exposed on open tundra would normally be consumed quickly by other carnivores or decomposers. The condition of the Duvanny Yar wolf indicates limited exposure prior to burial. Sediment analysis showed fine silts consistent with riverbank deposition. One hypothesis suggests the wolf may have fallen from a height or become trapped near unstable ground along the river's edge. Rapid coverage by collapsing sediment, followed by sustained sub-freezing temperatures, would have created the necessary conditions for preservation. Once encased in permafrost, microbial activity would have slowed dramatically. Computed tomography imaging revealed structural details of the skull and jaw musculature. The teeth showed heavy wear patterns, indicating a diet of tough fibrous tissue and bone. Genetic sampling confirmed that the wolf belonged to a Pleistocene lineage distinct from most modern Eurasian wolves. Its DNA contributes to a broader understanding of how wolf populations evolved and diversified during periods of climatic fluctuation. The presence of a well-preserved apex predator strengthens a pattern observed across Arctic discoveries. Mammoths, bison, rhinoceroses, horses, cave lions, and now wolves appear in frozen contexts with varying degrees of soft tissue retention. Preservation is not confined to herbivores or to a single ecological niche. It spans trophic levels. Ice core records from Greenland and Siberia show that the late Pleistocene climate was characterized by abrupt temperature oscillations. While these changes unfolded over years to decades, they demonstrate that environmental stability was not constant. In regions where permafrost intersected with active river systems, localized burial events combined with cold could halt decomposition swiftly. Standing before the preserved head of the Duvanny Yar, wolf compresses the distance between predator and present. The curvature of the canines resembles that of a modern gray wolf. The alignment of the muzzle and skull evokes familiarity. Yet this animal hunted mammoth calves and steppe bison across grasslands that no longer exist. The wolf's preservation does not demand a global catastrophe. Rapid burial and sustained freezing provide sufficient explanation. But its condition reinforces the same underlying observation. Across Siberia, multiple species occupying different ecological roles were sealed into frozen ground with biological detail intact. If a top predator could be buried and stabilized before scavengers and bacteria erased its form, then the environmental conditions responsible were efficient and decisive. And as erosion continues to expose permafrost cliffs along Arctic rivers, predators and prey alike are re-emerging from a landscape that once froze them in place. The blue babe steppe bison. In 1979, near Fairbanks, Alaska, gold miners working through frozen ground uncovered a dark mass embedded in permafrost. At first, it appeared to be a tangle of roots or a patch of stained soil. As the surrounding ice was chipped away, the shape resolved into hide. Beneath it lay the nearly complete carcass of a steppe bison, later nicknamed Blue Babe because of the bluish sheen cast across its skin by mineral staining in the frozen sediment. It was dated to approximately 36,000 years before present. Unlike fragmentary remains common in Pleistocene deposits, Blue Babe retained large sections of muscle, skin, and connective tissue. The head was present. Portions of the neck and shoulder remained intact. One foreleg was preserved with defined muscle fibers still visible beneath the hide. The carcass weighed over 1,000 pounds at recovery, despite the loss of some tissue over time. This was not a skeleton surrounded by impressions of flesh. It was flesh. Detailed examination revealed evidence of predation. Tooth marks consistent with Ice Age lions were identified on the hide and bone. Claw scoring suggested that the animal had been attacked and partially consumed before burial. The sequence inferred by researchers was direct. The bison was killed or scavenged, exposed briefly, then rapidly covered by sediment that later froze solid. The presence of predator marks alongside preserved tissue indicates that burial occurred before the carcass could be completely dismantled. Under ordinary Arctic conditions, a carcass left on the tundra would be quickly consumed by wolves, birds, and other scavengers. Even in cold climates, freeze-thaw cycles and microbial processes degrade soft tissue over time. For Blue Babe's muscle and hide to remain recognizable, freezing must have stabilized the remains before decomposition advanced beyond a certain threshold. The sediment surrounding the body consisted of fine silt and loess, typical of wind-blown and river-deposited material in Pleistocene Alaska. Such sediments can accumulate rapidly during dust storms or seasonal flooding. If the carcass lay within a low-lying area or depression, it could have been covered quickly by sediment and subsequently locked into permafrost as temperatures remained below freezing for extended intervals. Scientific analysis of the preserved tissue allowed study of muscle fiber structure, hair morphology, and even isotopic signatures reflecting diet. Collagen extracted from the hide provided additional confirmation of age and environmental conditions. Unlike mineralized fossils, preserved soft tissue offers direct biochemical information about Ice Age ecosystems. Blue Babe's condition reinforces a broader observation across Arctic and subarctic regions. Large herbivores were sometimes killed, partially consumed, and then sealed into frozen ground before natural decay could erase their anatomy. The freezing that followed did not mummify gradually over centuries. It arrested decomposition at a definable stage. Climate records indicate that the late Pleistocene environment of Alaska and Siberia was characterized by strong seasonal contrasts and episodic sediment movement. In such landscapes, the combination of predation, rapid burial, and sustained cold could produce preservation conditions rarely seen elsewhere. When displayed after conservation, Blue Babe did not resemble an abstract fossil reconstruction. The hide still draped over muscle contours. The impression was biological rather than geological. It conveyed immediacy. If a 2,000-pound Ice Age bison could be attacked by predators, buried before being fully consumed, and then frozen in place for more than 30,000 years, then the transition from life to permanent ice required only a narrow window of time. Across Siberia and Alaska, similar windows appear again and again in the geological record. And as thawing permafrost continues to expose these sealed moments, the frozen ground of the North remains not only a record of extinction, but a chronicle of how swiftly environments can close over the living. Conclusion. Across Siberia and Alaska, the pattern is consistent and physical. Mammoths with skin still attached, calves sealed before digestion completed, predators preserved with teeth and whiskers intact, a cave bear emerging with claws and fur still visible, a bison partially consumed, then frozen before nature could finish its work. These are not isolated curiosities. They are repeated biological interruptions recorded in ice. Permafrost does not fossilize in the traditional sense. It suspends. When burial occurs quickly and temperatures remain below freezing, decomposition slows to a fraction of its normal pace. Muscle retains structure. Collagen persists. In rare conditions, even delicate tissue survives long enough to be studied tens of thousands of years later. The late Pleistocene climate was dynamic, with oscillations capable of shifting environmental conditions in relatively short intervals. Within unstable riverbanks, floodplains, and collapsing slopes, rapid burial followed by sustained cold created natural cryogenic chambers. What makes these discoveries increasingly relevant is not how they were frozen, but how they are returning. Arctic temperatures are rising faster than the global average. As permafrost thaws, long-sealed biological material is being exposed to modern ecosystems. Scientists now study these remains for genetic, ecological, and climatic insight. Each specimen expands understanding of Ice Age environments and extinction processes. The ice once acted as a stabilizing archive. Now that archive is destabilizing. The animals preserved within it are reminders of how swiftly conditions can shift and how decisively environments can close over the living. The past was locked by cold. The present is unlocking it.